So let's talk about language models before we talk about protein language models. In general, in language models, the idea is, let's say I have a set of words. Let's say I have a bunch of sentences, and I'm trying to come up with a model that's able to predict this word. So whenever I have a blank in a sentence, the goal is to predict what word would be best suitable for that spot. Or better yet, let's say I have a whole paragraph, and then my goal is to predict what this entire next sentence should be. What are some types of information we could gather from the surrounding context that helps in the case of this word? Okay, so maybe this specific word is sort of the biggest hint as to the blank. Anything else? So maybe we're expecting a noun here based on grammar. Do you think that's learnable? Is that a learnable rule for the English language? Okay. And is this relationship learnable between color and maybe some color has to go here, matching with this description? Okay. So maybe give me possibility. Okay. So now let's get out of this space and consider, is it similar to this? Okay, what should go here? An animal, such as, so let's say I have something similar to this sentence. What do I do then? It has to be a category. What does the category define? Drink? Seems reasonable. So now let's expand this whole thing. Obviously, this is a very specific sentence structure. My favorite blank is blank. So let's think of this. We have access to a bunch of sentences. Let's say every Google search that's considered a full sentence we have access to. Let's say everything in the dictionary we have access to in terms of as far as words go. And then let's also say we have access to every single book written. And maybe that's our data set to learn how to fill in blanks in general. So maybe it's not as similar in structure to this, but we know the English grammar, and we also know that we have to fill in the blank and predict as accurately as possible. So if I predicted water here, it wouldn't be very accurate, right? Because people wouldn't say that. So that could help us figure out what kind of loss we need for either word prediction or sentence prediction. And then do you think if we have a model that has learned on all known forms of text or all accessible forms of text, it would be possible to figure out how to write sentences from scratch? Can you generate text from scratch? Okay, so let's say that I'm interested in writing a paragraph that's about cats in general. Do you think I would need some type of context to maybe steer the paragraph? So maybe it is the drinking or water drinking habit of cats. So I can contextualize a little bit, and then maybe that's my input to a model for text generation, and then I end up with a whole paragraph that's somewhat informative. And so what we're going to discuss is the relationship between sort of all categories of different things that you can learn on and how you can relate it to, let's say, these different possibilities, and then coming all together. Well, do we know what types of characters are possible to come after M, at least in English? Is it likely to see another M if M is the starting character at 0 position? Maybe not. Do we know when we need spaces? Is that predictable? So let's say that you're going to fill in the blank, and then for each of these positions, so for this word, the character 1, 2, 3, 4, 5, and 6 are known. So is it easier to figure out these two characters, for example. And so over time, you could collect some information as to, first of all, what character is possible to appear after this one, what word is likely to start a sentence, given you have a bunch of sentences, and also sort of if you're specifically trying to fill in a blank, what could go there? If you have bigger context for the text generation, what makes sense? And then putting all of this together, what people have done, which is really remarkable, and it's sort of been around for a while now, they have developed language models that are able to sort of do this and do it quite accurately, to the point we can search and generate based on this context. So it's almost like we can construct a whole sort of informative text based on something that we want to learn about. So what's an example of this? Some of you have been interacting with ChatGPT. Okay, so let's pause here. Obviously, from the title of this talk, you can see it's language models for proteins, or protein language models, PLMs, whatever. How do we apply this type of logic to proteins? Let's discuss that. So in terms of text, or let's say a string of characters, we can see some patterns. They have some type of position, and we know perhaps after first M starting a sentence, we're not going to have another M, etc., etc. We know that from this word, we can pick up some context clues for this word, and then altogether, we can come up with a context for an entire paragraph. If you are asked to summarize a paragraph into maybe 3 keywords, you can do that, right? Humans do that, so they write abstracts and then they have keywords identifying what that paragraph explains to get sort of the correct audience for it. And so, okay, well, that seems really powerful, but how can we do this for proteins? How do you pick up protein pattern that's maybe similar to words and sentences in some sense? Maybe we're interested in speaking the language of proteins, and we can have the protein sequence be sort of the sentence, or it can be what we're trying to construct based on some clue. And in terms of the clue, instead of maybe water intake of cats, we're interested in, well, if I wanted to design antibodies and I give you the category antibody, and maybe that's sort of an annotation associated with producing or generating a protein string, let's call that the protein sequence, how can you do that? Do you think there is some context that can be determined by the type of protein? Do protein families constrict what type of sequences are allowed? Is there a relationship between function and protein sequence? Is there a relationship between protein function and protein family? Right, so based on some type of classification for protein, let's say families or functional families, and then their sequences, we can figure out some other learnable parameters and then apply language models or advances in language models to protein design and see what happens. So we're going to talk specifically about a method that was produced in the Gray Lab by Jeff Ruffalo, and it is called IgLM. So what is Ig? Immunoglobulin. Okay, what is that? An antibody. And what is LM? Language model. Okay, so this is a language model specifically for antibodies. Let's talk about it. So this talk was given at last year's Summer RosettaCon, and I'm just going to be presenting on these slides. So the title of the talk was Generative Language Modeling for Antibody Design, and this might actually be recorded from last summer. I'll see if I can find the link to the recording so you can watch it. There might be some interesting talks there. Same for protein MPN, actually, was presented at Summer RosettaCon. So I'll see if I can send you those. Okay, let's talk about it. So we're going to need some learnable database. So if we're learning this from a bunch of books, let's say we have a whole library, a collection of text, then we can learn about text. So for protein, for protein design, we're going to need sequence libraries so that we can figure out what pattern is possible to learn at all. And so in sort of putting together different antigen specific antibody libraries, people will go through experimental processes like yeast display, so you can select for specific antibodies and then construct and refine on a library that you're interested in using. So in terms of antibody format, there are some differences as to which type of system people would study. So today, we're going to be specifically talking about the nanobody here, as well as this single chain FV. So if we had a model that's able to generate new sequences for the library that we can test and then sort of put into any of these antibody formats, it would surely be interesting, because what do antibodies do? So maybe it leads to a good thing after it has bound a bad thing. Okay, so would you say it's important to have antibody function in cells? Okay, so we're going to start off with antibody-specific generative language model, and then you're also going to do a demo on the rest of this as well. So let's just start talking about the model and how it was put together. So we know from the let's say our immune system, we know that there's going to be a snapshot of antibody sequences that a person is reproducing, and based on that, we can identify common antibodies as well as those specific to antigen. So if we wanted to have some relationship between antigen and antibody, we would need to pick the ones that are specific to their antigens and are also sort of functional inside some biological system. And based on this collected data, we can actually study and put all of our protein data into a format that's learnable. So observed antibody space, or OAS, is the database, and from training data set that is constructed with OAS, IgLM was built to essentially go from some type of model input. So in here you can identify different types of protein sequences as well as what you're interested in seeing. So that might be species. Earlier, Amy was mentioning that you might be interested in just saying something like the protein type, so maybe its function. But in here, our input is going to be antibody-specific, so antibodies have heavy and light chains. So from your from your pack and relax example with the antibody, you got to see an antibody structure and the CDR loops for the heavy chain. So this is describing the heavy chain. And then you can describe some type of species. So besides human, what might be some species that have antibodies enough that it was learnable. So it could be mouse antibody. And then you might describe some initial position. So we're going to talk about this sequence right here. And then what does it mean to mask a part of the sequence? It's hidden. So maybe you're going to give some of the positions or fix a certain portion of the sequence in the structure, but figure out what the rest might be. Did you do something like this maybe yesterday? Did anybody fix a part of the sequence going into protein MPNN? Right? And so in that case, you can think of it as the portion that's hidden is the portion that gets designed, which makes sense. And so with that, we can have even more control over what actually gets to be inputted in our model. And then IgLM is our model. And then we end up with a probability distribution over the 20 amino acids. And you got to take a look at this yesterday as well. If you remember, you looked at this at different positions. So you can sort of relate to this idea. So in here, which amino acid has the highest likelihood according to IgLM, and which one has one of the lowest likelihoods according to IgLM. So the authors were able to get 558 million natural antibody sequences. Does that number seem like enough for learning on antibodies? So did you notice about sort of everything that wasn't a CDR loop when you were doing that exercise? They seemed somewhat unchanged when you were going through the protocol. So normally people don't design the region besides CDR loops because of immunogenicity once again. And so if we consider that and that we're interested in sort of infilling loops, then it becomes, well, maybe this is very sufficient because we know what we need to change biologically speaking. And so for the part of the context that is sort of what we're interested in filling in, maybe it is possible and maybe we can do this very accurately. Okay. So based on clustering this and sort of analyzing it, so this is the number of sequences per different types of species. We said human, mouse, but there's also a camel, so rat, rabbit, and also with different types of heavy and light chains. Do you notice anything about heavy versus light chains here? So which species have both heavy and light chains? So we're going to discuss this in the exercise coming up, but previously you have looked at heavy and light chain sort of in complex together, just to contrast that. And so we talked about sampling temperature in a different lecture, but in this case, once again, increasing the sampling temperature will lead to sort of if it's very high, it will lead to degradation of generated sequences, so it becomes very unrealistic. Looking from here, let's just say at a lower temperature, when we sample some loop here, and then we increase the temperature, we increase temperature, what do you observe around here? What is the difference between this low temperature and this sort of moderately high temperature? What happens to the protein structure? So it starts forming some other secondary structure here, or almost breaking some secondary structure. And then what happens here? So I heard disordered. This is disordered. And what happens by the time you reach 1.7? Okay, so it kind of unfolds. So whatever you ended up designing in that position is very low likelihood in general to fold, right? So it doesn't even look like an antibody. It doesn't look like a well-folded protein at all. And so once again, sampling temperature is something you would have to tune and sort of figure out where to stop. So why do we want to increase it at all? Because this seems like the best sort of structure, but why would you want to go slightly higher at all if we're doing so well, structurally speaking, in this lower temperature sampling strategy? So as we go towards zero, our answer is basically converging. So we're just getting one very likely sequence according to IgLM. And since it has learned on real antibodies, if we really want to get away from sort of the existing space for what is allowed at those positions, then we might want to reach for some diversity and go higher up in the sampling temperature. All right, so now let's talk about controllable generation of full-length sequences. So whenever we generate sequences, we're interested in figuring out if we want to, just like this example, if we want to write the whole paragraph or parts of the paragraph. So if we have one sentence that we want to keep, but sort of write around it, maybe you have, let's say, a part of your abstract, but you want to sit down and think about exactly how the rest of it comes together. Maybe you're missing the introduction line, maybe you're missing the conclusion line, right? And so let's think in terms of sequences. So once again, our conditioning might be based on heavy or light chains, so which type of chain and which species to design this for. How much variation do you think there is between human, camel, and mouse antibodies in terms of sequences? Do you think you could use a rat antibody in humans? Do you think you could use a human antibody in rats? How about camel? Can you use a camel antibody in humans? Can you use an antibody that was made by a model that doesn't look like any of these species in humans? Okay, so you'll have a chance to explore the differences between species in terms of both sequence structure and structure prediction type analysis. I would also add that it'll be interesting to fix different positions and see what happens in terms of different species. But so let's just talk about, based on these conditioning tags, we can feed into IgLM, we can come up with generated sequences that contain that chain, as well as according to IgLM, it belongs in that species category, and then we have our full sequence. So based on the sequences in OAS, which is once again where we have the data used for training, the number of residues that are missing from the N terminal is going to have some influence as to what is learnable. So this was a problem for sort of continuing with this model in the beginning, because you had to figure out, well, for a percentage, so let's say around 16.2% of sequences starting here were missing the first residue. And so it becomes harder to learn, let's say that your starting word or maybe starting 3 words are missing. It becomes a little bit harder to predict and put together the rest of the paragraph. And so applying this to antibodies, what they decide to do is either give a prompt where you can sort of give it the initial 3 residues and then allow for complete sequence generation so that the sequence length was sort of reaching what we would expect it to reach with the prompt being in orange, versus without that prompt, it was quite a bit shorter, at least for a distribution over here. And then this is in the heavy chain, this is in the light chain. So once again, no prompt would mean that we give how many initial residues as a starting point? 0. And then with the prompt, what does it mean for us? How many residues do we provide as an initial starting point? 3. And so let's take a look at the sampling temperature and how it controls the number of mutations. So there's a direct relationship between the number of possible mutations and also the temperature, because as we sort of increase the temperature, we're going to allow for a little bit more chaos, and maybe this controlled chaos allows us to step outside of some sequence identity space. So let's say that we're quite identical to all other sequences for humans in the heavy chain, and then let's say specifically for this gene, and we're interested in stepping outside of that, so we can lower the identity by increasing the temperature. This is very similar to the percent recovery of sequences, which is a concept that we covered yesterday. And so its relationship to temperature is once again observable in terms of mutational load. Okay, so let's talk about infilling. And okay, what are CDR loops once again? Somebody tell us what they are. Maybe what is the abbreviation for? It was complementary determining regions, that's right. And then why do we care about CDR loops? So they are essentially the binding loops for antibodies, and they recognize the binding partner. Okay, and then if we have, let's say, a heavy chain, how many CDR loops are there in the heavy chain? 3. And then how many for a human light chain? Also 3. Okay. Okay, so now let's take a look at this. Maybe we have these different types of drugs. So these are going to be different types of antibodies. And what is this color here representing? Do you remember from previous slide? We have a heavier light chain, the species, and then what do you think this is right here? Okay, so if you look here, we're generating a sequence, right? Starting from any sequence at all here, or just some tags that we're conditioning on the tags. And then now we're giving some sequence, but we're interested in infilling essentially the H3 loop. So H3 loop is in which chain? The heavy chain. So it's the third loop in the heavy chain. So the goal here is, well, if you remove the CDRH3 loop for each of these different antibodies and then generate a number of sequences using IgLM, and based on some sampling strategy shown here, where you're changing the temperature and the nucleus probability, what types of structures can be predicted based on the designed sequences. So IgLM will design sequences for the missing H3 loop based on this sampling strategy, and then using a different model. So you can think of this as, it's similar to AlphaFold, but it was developed for antibody structure prediction. And so given these new sequences filled in by IgLM, can you predict structures that look like antibodies for it? So for all of these drugs, when you make changes to the H3 loop, if you are successful in designing and in filling the correct sequence, or one that should have very high likelihood and would be successful experimentally speaking, do you think that the structure will look pretty nice, right? So for our results, maybe we're analyzing any irregularity in structure. So if it looks unfolded, that's not good. But maybe we can evaluate this method by making a bunch of sequences and then predicting structures for it, and then seeing, well, do the structures for where we didn't design, so besides the H3 loop, does it align well with the structures for each of these antibodies, right? Do we have the ground truth structure for all antibodies? Is it possible we have some sequences that don't have paired antibody structures that were experimentally determined, right? So that's also a possibility. And this sort of drives the point for why you would want to have an antibody-specific structure prediction method. And so let's take a look at some of these. In this case, they're infilling it for one drug, and this is done a bunch of times. And based on that, they're analyzing the H3 loop length. And then, so shown here is just one drug where you have infilled the H3 loop a bunch of times, and then the parent H3, so the real H3 is in black. What are some observations on just this part of the figure right here? What are some differences between the turquoise, which was designed by IgLM, versus the parent, which is in black? So the infilling is for one H3 loop, but it is done a bunch of times, and we're sort of showing them overlaid on top of each other. But you only fill in the H3 loop once. Okay, it's longer. So how does that help us? Is that a good thing? Can you maybe design this antibody for a slightly different target and make this H3 loop as long as it needs to be to have that surface complementarity to its binding partner? Okay, so you could see it being helpful. And then do you see some variation in terms of length? So for this shown right here is going to be the second bar right here. So we actually have some variation in terms of the H3 length. I don't know off the top of my head what the parent length is, but essentially you could sample so much so that you're getting to very short segment here. Is 6 pretty short for H3 loop length? That sounds pretty short. So in here, we have all sorts of lengths, and we could see it sort of being useful to have the ability to design longer H3 loops. Okay, so now let's talk about likelihood. So IgLM is able to calculate likelihoods based on sequences. So the whole goal of this is to see, well, does it look like a human antibody, or does it look like a different species according to IgLM? Why do we care about sequences that look like human antibodies? Okay, it's less likely to be immunogenic, so maybe it doesn't have adverse effects. Any other reason? Okay, so for design specifically, let's say that we are coming up with a new library of human antibodies, and we're going to test the most successful ones. Do we have the capacity to test for the entire library, right? Experiments take a while. And so maybe we can figure out some ways to use IgLM to develop sort of a screening process computationally and then have higher experimental success. So in this case, you're seeing the likelihood of having one of the sequences that belongs to either of these categories in either the heavy chain or the light chain. And then this is plotted based on that log likelihood. So what do you see? So the human one is going to be in blue, and then humanized is in orange, and then you're going to have humanized chimeric in green, and then you have a whole bunch that are non-human in the rest of the colors. So discuss with your table for maybe just 30 seconds. How do you interpret this figure? So maybe we see the human antibodies separate quite well, so they sort of cluster and blob together up here. And then for the rest of them, it doesn't seem to be as distinguishing. And so my question to you is, well, you see these orange points up here in the human cluster. So we know orange is for humanized antibodies. So what does it tell us about that orange sequence up there? Is it a mistake? Is it by mistake that it has that likelihood? It's so high with the human antibodies? Yeah. So maybe this humanized antibody would work just as well as a human antibody, such that it's very difficult to actually discriminate between them, even with IgLM, because it looks really good. So that's a good thing. And in here, based on this type of likelihood analysis, we can analyze whether, so we can sort of compare different sequences and see whether we're interested in using them for further rounds of design or for testing because they look human enough for us. And so IgLM is particularly successful at this discriminative task. Okay, and so to summarize, this generative language model for antibody design is able to generate sequences conditioned on certain control tags, such as the species, as well as what kind of chain it belongs to, and then we're going to have a set of sequences. It's also able to do some sequence generation given a prompt. So this prompt might be a set of initial residues. This is especially useful to complete or give us a head start, given that in some sequences in OAS, we don't have the first 3 residues. So it sort of compensates for that. And we can also have targeted CDR loop diversification. So we can generate specifically a chosen loop in the CDR and then keep the rest of the framework residues constant. And then for this designed region, we're going to have a bunch of different loops. So this would diversify our entire binding region and make it more successful to perhaps a different target, or maybe for us to increase affinity or specificity to the already existing target. And so this is the power of using this method specifically. And we're going to get started with a demonstration using IgLM. Okay, so let's get started. And this is the end of the recording.